So this is nonlinear self-adapting wave patterns. Try to explain what that means and what that's all about. This is work that was done with Herbie Levine from Rice currently, appeared last year there. Also had questions about what I should talk about, and sort of perverse, I probably should talk about fracture because that's when I first met Jay at Aspen way back when. I don't even know, 80, 89 maybe, 90, something like that.
When my car blew up.
But this is, first of all, because Jay worked on wave patterns of other kinds, Faraday waves and things like that, and patterns and stuff like that, so hearkening back to the good old days of yesteryear. And also this was work done in Aspen, so it reminded me of Jay, so that's by way of apologies. All right, so let's talk about simple things first that that everybody understands. First of all, the most simple pattern, the classic pattern, is Turing patterns. When you start out from a uniform state, and Turing discovered back in the 1950s, right? Yes, that you had bifurcations from a uniform state to stationary spatial oscillations. So that he wrote down a model that when you turn some parameter, you turn some crank, the system originally has a uniform state. Always the system always has a solution which is constant in space, but at some point it develops a new solution which is oscillating in space, stationary in time. So when you do the linear stability analysis, you find out that there's a real instability. In other words, the imaginary part of the growth rate is 0. There's supposed to be an imaginary part there. And at some finite k, some finite wave vector, all right, which sets the wavelength of the pattern. And so above threshold, at the instability, there's exactly one unstable mode, so you get exactly one wavelength. But then you go slightly above threshold, you have a small band of wavelengths. But still in all, basically, there's the length scale of the pattern determined by the k that went unstable. And so you get these wave solutions, these solutions oscillating in space. And to get this requires sort of the minimum way to get this is either two fields with different diffusion constants or a higher order equation like the Swift-Hohenberg equation, some fourth order equation. All right. Okay. And there's another class of instabilities called hot bifurcations, which are instabilities at k equals 0 and pure imaginary. At the point of instability, the growth rate of the perturbation is pure imaginary. So below it, it's a small negative part with an imaginary part. Slightly above, it grows exponentially, but it also oscillates in time. All right? And in principle, there's a third class. The third class is one that is a traveling wave bifurcation. In other words, in principle, you can imagine a situation where the mode that goes unstable has k not equal to 0 and imaginary omega not equal to 0. So what's strange is that as opposed to the other two cases, you look around, there's no standard model, if you want to explain to a beginning student, let's write down a simple model that has this kind of instability. Well, for Turing, Turing gave us one example. There's the Brusselator is another example. There are lots of examples of very simple models which have Turing instabilities. And in fact, most of those models, if you go to a different range of parameters, they have hot bifurcations. So those are easy, and there's a classic and worked out, and all is well and good. But there didn't seem to be a simple example of a traveling wave bifurcation. So, and since we were sitting there trying to write a book about explaining all this stuff before all this stuff gets forgotten, and so we were looking around and said, okay, but we know there are systems that have traveling wave, exhibit traveling wave bifurcation. So let's just go to there and look at them. Somebody must have worked out the theory for them, and we'll just copy it down and maybe simplify it a little bit so that it'll be easier to explain to a student. But, all right. So, but in fact, there's a reason why there's no simple standard textbook example, because it's actually quite hard to get this thing. Because in particular, it requires work to guarantee that the k equals 0 mode remains stable. Because because usually what happens in diffusive systems is that the bigger k is, the more stable it is. So if you can imagine that the system is unstable at k equals k naught, well then it's surely unstable at k equals 0. And so the first instability is the Hopf instability. And so how do you get a system where the first instability is in fact at k not equal to 0 and imaginary? So we looked at an experimental system where we knew that there was a traveling wave instability, a system that's a biological system called the Min system. So now I have to explain a little bit about the Min system. The biological challenge that the Min system is designed to solve is you have bacteria like E. coli, and you want them to divide more or less symmetrically. All right? You want them to, they grow and then at some point they split, and you want them more or less to have equal amounts of stuff in both sides. All right? And the biologists went ahead and they actually found that there's a set of proteins that are responsible for guaranteeing that E. coli split more or less symmetrically, divide more or less symmetrically. And in fact, they discovered that if you knock these out, then in fact what you get is you get very asymmetric divisions, and in fact you get what they call mini cells which are DNA free. Because what happens is you get a little pinch off of stuff and there's no room for the DNA, and so that's very bad biologically, obviously. And so that's why they're called Min, because when you have a mutation or whatever, you knock these things out, then you get mini cells. So these are the Min proteins. So this is the Min system, and there are three of them, Min, C, D, and E. All right. And so this system has been studied experimentally at great length. It's a very interesting system for a biologist and physicists. So it's been found dynamically that there's a standing wave oscillation set up in the cell with a node at the cell center. So you get this sloshing, you get this kind of oscillation. And so if you look in the middle, so you can tell where the middle is, because the middle is this thing that's not oscillating. All right, the ends are, the amount of Min at the ends is oscillating, and at the center it isn't. So the cell now knows where the center is. All right, and so the node has on average the lowest amount of Min D, and that causes a minimum of a Min C at the node, and that's therefore that's involved in setting up the physical mechanisms that cause the cell to split. Right? Min C inhibits the formation of what's called the division septum. There's this, you get this thing so the cell actually grows new material and makes a new cell wall in the middle, and so the septum then forms at the center. And so but the oscillations actually are driven by Min D and Min E, and Min C just responds to the oscillations in the Min D, E. So it's enough to have Min D and E to get these oscillations, this standing wave, and then Min C is sort of, is what uses that and translates that downstream and interfaces with the rest of the biology. So, all right. So this Min DE dynamics has been studied, and it turns out that Min D and E exist in the cytosol, in the interior of the cell. And we're going to call the concentrations of Min D and Min E in the cytosol with capital letters. So the C sub capital D and C sub capital E refer to the Min C and the Min D and Min E in the cytosol, in the cell volume. And also, Min D can exist on the cell membrane. So we're going to call that with the small d. So C sub small d is the Min D that's on the membrane and not in the cell, but on the envelope. Now, Min E by itself does not exist on the cell membrane. It only exists when combined to min D. What happens is the min E sees this min D on the cell membrane and can attach to it and form a complex. So that the concentration of that stuff is called C sub DE, small letters because it's on the membrane, DE because it's a complex between D and E. Okay. All right. And then these stuff don't stay on the membrane. They eventually leave the membrane, go back into the cell and then they split up. The complex, when it goes in, splits up. All right. So, and then all types diffuse, both on the cytosol and on the membrane. And for simplicity, we're going to think about a one-dimensional system because, we're physicists. So we have one-dimensional cells. So the equations you write down, these are not our equations. These are a standard set of equations written down by Luce et al. In a paper in Science in 2008. There are 4 equations for the 4 concentrations. All right? And so if you think about the change of D in the cytosol, well, it's because the DE complex on the membrane split up into DE in the cytosol when I'm bound from the membrane. Then D goes onto the membrane, so that takes away from the capital D in the cytosol, and then there's diffusion. All right, and then similarly, when you look at the equation for E in the cytosol, it's DE decomposing, and then there's also E leaving the cytosol when it joins with a D that's on the membrane, and then there's diffusion, and then similarly for the next 2. And the key point about this is in this model, the total amount of C, of D and E, of min D and min E is conserved in its various forms. So the total amount of min D is the D that's in the cytosol, the D that's on the membrane, and the DE, the complexes of DE that are on the membrane. Similarly, if you think about min E, that's composed of the E in the cytosol and the DE complexes on the membrane. And if you think about this, if you look at this equation, and this equation, you find that all the reaction terms vanish. And therefore, the total amount of D is conserved. And similarly, if you add this equation and this equation, this term cancels that term, this term cancels that term, and the total amount of E is conserved. All right? So that's just a feature of the model. All right. And then you can write, then in this paper by Luce, they write down what the reaction rates are. They're nonlinear, except for the desorption of the DE from the unbinding of the DE to the D and E in the cytosol, but the other ones are nonlinear. The other point to notice is that diffusion on the membrane is obviously much slower than diffusion in the bulk. And and then these in this paper they have, guesstimates, measurements, some mixture of measurements, guesstimates, whatever, for all these parameters. So there's a model. Very nice. And in particular, you can solve for there's a uniform equilibrium solution, right, with a certain amount of stuff on the membrane, a certain amount of stuff in the cytosol, and nothing depending on space. And and now in principle, so what they did in this paper is they did simulations. That's because we live in a modern world and it's cheap to do simulations. You have a bunch of equations, you throw them on MATLAB, and you say, MATLAB, please simulate this for me. And even people who are biologically oriented can manage to do that. And you get pretty pictures of oscillations. And they're very happy, right? And then, and what they didn't do, amazingly enough, from my perspective, is they never did this stability analysis. They never actually calculated the stability analysis. Why didn't they do it? Because to even solve the steady state equations is a nonlinear problem, right? You have 4 equations, all of them are nonlinear, you have to solve them. There's no way to write down an analytic solution for that. You just have to solve it numerically. And now, what's the point of doing, and now you have to do the linear stability analysis. You have basically involved the zeros of an 8th order polynomial, right, whose coefficients depend on this actual solution of the steady state equation, which you only have numerically in any case, right? So it's a big numerical mess, so why bother doing it? Right? What you would learn? That was, I guess, the feeling. So they didn't do it. All right, but we wanted to explain, to see this bifurcation. So we said, since they see waves, there must be a bifurcation. Let's go find it. So we did this stability analysis. So we just did it numerically, because it's not that big a mess. I mean, it's just a bunch of equations. You write it down and you get a matrix 8 by 8, and you get the eigenvalues. What could be hard, right? And this is the growth rate and the frequency of the least stable eigenmode of this 8 by 8 system. Okay? And as a function of k, you look for perturbations, since the bare state is translation invariant, so obviously all eigenmodes can be labeled by Fourier number k, and so you plug in k, and you just compute, as a function of k, what is the growth rate and frequency of these modes. And lo and behold, so this is for two different values of d on the membrane. Let's just focus for the moment on the dark blue. So this is the real part of the growth rate, and this is the imaginary part of the growth rate. And you look at it, and something jumps. First of all, you see that it's unstable. Very good. There was an instability of the uniform state. That's why you get waves. Well, victory. Okay, but then you look at it again, and you say, there's something funny here. The growth rate goes to 0 at k equals 0. Yeah, well, right. So people who are smart would have said that immediately. People like me who are slightly less intelligent, once we look at the graph, then we have the brilliant insight that the reason it's 0 at 0 is because of conservation laws. Right? It's just a question of I like to cheat and look at the answer, and then I can pretend that I'm smart and understand why. All right. So what do we get? And then the modes are, except at k equals 0, the modes are complex for all k. So here we found, yeah.
I didn't really understand what you meant by membrane.
It's so it.
Homogeneous? Is it a homogeneous?
Okay, so we
inside the cell, or is it the inner surface?
well, imagine the cell originally. Imagine the cell is some ellipse, right? And then we're going to imagine that it's much longer than it is wide, so that the variations across the cell can be ignored. And so now we just have a one-dimensional. We have a
So it's a surface between the.
right. So okay. So we have good things. We've got complex mode. We have an instability at finite k. And we have, right? And k equals 0 is not unstable. So there's no hot bifurcation. So, all right. So now once you do that, all right, there's the picture in miniature. All right. We can things actually are very simple around here. Because around here, for small k, there are basically two modes here. There are, you see, because we have this complex mode, that means there's a plus I omega and a minus I omega. So there are two modes. The other modes are much more stable. We can throw them away. And basically, we can use degenerate perturbation theory to calculate what happens, because we know the exact answer of the problem at k equals 0. We can do analytics, right? Which you would have thought, without thinking that it was a hopeless task. Now it's a very simple task. We have 2x2 matrices, something that a human being can actually manage. So you write down the stability matrix, and yeah, the entries are numerical because they depend on the steady state solution, but the structure it's proportional to, the matrix is proportional to k squared, because when k equals 0, both modes are 0. So the matrix is just 0. And obviously, therefore, they're proportional to the diffusion constants, both desol, and there are 4 diffusion constants, but to make things simple, we'll imagine that the 2 cytosol diffusion constants have one value and the membrane have another. You can do the more complicated case, but it's just easier to think about what's happening here. So you get things which are proportional to D in the cytosol, which is the big diffusion constant, and you get terms which are proportional to D membrane, which is the small diffusion constant on the membrane. All right? And now the eigenvalues, you can calculate the eigenvalues of this 2 by 2 matrix, right? And the eigenvalues are clearly proportional to k squared, and you can actually figure out when they're complex. And they're complex if d sol over d mem is bigger than 17.8 for the particular values of the parameters of the reaction rates and stuff like that. So there's actually, as long as the cytosol diffusion is much greater than the membrane diffusion, which it in fact is, then in fact you get complex modes. All right. So for d sol much, much bigger than d membrane, the initial growth rate depends on d sol, and what happens is this eventual restabilization depends on d membrane. So when d membrane is big, then it happens here. When d membrane gets smaller, the eventual restabilization happens at much larger length scales, but it always happens. All right, so now we've done the linear stability analysis, and now what does it mean? Because it's in fact not what we were expecting, and not what the words, if you read these papers, right, they saw these traveling waves and they talked about it as if it was a Turing kind of instability. In other words, that there was a specific wavelength associated with the instability. But in fact, there is not a specific wavelength, because the minute you have unstable modes, you have, perforce, an entire window. You have a family of solutions because it goes all the way to k equals 0. In other words, it goes the lengths extending out to infinity. There's no unique length scale. So the two conservation laws, first of all, give us a marginal pair of modes at k equals 0, and they naturally mix. And the reason you get complex things is because you take two things and you mix them. It's not so, it's generic that they're going to give you complex things because these are not symmetric matrices. So you should expect that there are regions of parameter space where you get a complex conjugate pairs. There's no hop, global oscillation to worry about because we've guaranteed, because of the conservation laws, that at k equals 0, there is no instability. So there's no hop to worry about. All right. The fact that the unstable band always goes down to k equals 0 means that there are solutions at all length scales greater than some minimum. All right. And which is very different from this Turing instability with its fixed length scale, and very reminiscent of things that happened back in my troubled past when we studied things like dendritic growth and viscous fingering, where again there was a single real unstable mode, also because of a symmetry.
The computer's about to shut down.
Oh,
No power.
for no power. Yeah. Give me my bag.
You have to finish it anyhow.
There's enough time.
All right. Okay. All right. Very good. See how perfectly we timed this. Okay. In viscous fingering, the length scale, there was one real unstable mode set by symmetry, and the length scale of the pattern, therefore, is set by the system size, not by the stability spectrum. And so what we expect is traveling wave solutions for all L bigger than some critical L, and the amplitude should grow like the square root of L minus LC. All right. And in fact, you can find them. You have, if you put on periodic boundary conditions, then you can find these traveling wave solutions, and they exist, and everything is hunky-dory. Okay. Now, and then when you increase L, the amplitude increases. All is well and good. You get all other kinds of interesting things, because now you have multiple solutions. You can construct an L equals 20 solution, a solution in a box of L equals 20, either by looking directly for an L equals 20 solution, or you take two L equals 10 solutions and just tie them together. That's exactly a periodic thing with L equals 20. And that solution turns out to be unstable to coarsening, just as what happens in Saffman-Taylor. If you turn out start out with two Saffman-Taylor fingers and let the system progress, they interact with each other, and any small perturbation causes them to interact with each other, and you get a single finger at the end of the day. That's what happened here. We started out with two L equals 10 solutions, and in the end, we get an L equals 20 thing. Okay. So now, of course, in a cell, you don't get traveling waves because the cell is finite. You get standing waves. So all that happens is the traveling waves get go over to standing waves, and here are the min oscillations that everybody knew about, but now we know why they occur at some deeper level. And the last point is the fact that the pattern has no intrinsic scale is in fact perhaps potentially crucial to the biology, because a cell doesn't come in a fixed size. Cells grow. The one thing we know about cells is they start out right after division, they're this size, and then they get bigger, bigger, bigger. Eventually they reach some other size, and they split again. So in fact, if you're trying to set up these oscillations, and all of the time the cell is growing, well, that should screw up these oscillations and change intrinsically the nature of these oscillations. Well, not in our system, because the system basically has no preferred length scale. It's perfectly happy. You tell it the cell size at any given moment, and it just adjusts itself automatically. So here, in this simulation, we let the cell grow exponentially in time with a doubling time of 50. We ran it to time 5,000, and the system just kept its structure all the while and just changed its length scale dynamically. All right, so just to wrap things up, the conservation laws that we have here eliminated the possibility of a Hopf instability. If you have one conservation law, then you get a real instability. If you have two conservation laws, then you have opens the way for a finite K, finite omega instability. The fact that you have no minimum unstable K gives you flexibility of size, gives you coarsening, and this self-adaptation. And happy birthday, Jay. Yes.
Concerning the difficulty of finding finite K instabilities.
Yeah.
Because in binary, in convection, or in Taylor-Fouet, or all these systems, there are plenty of finite K instabilities, and
but okay, they break. Yeah.
Oscillate.
They break. Okay, in like the Couette, right? You have a direction already of rotation.
No, I'm talking about in this direction.
So what?
Spiral waves.
No, I'm not talking about spiral waves. I'm talking about Taylor vortices. There's a K that's chosen, or convection, rather than convection, reflection symmetry. There's no breaking of the reflection symmetry, and you have a finite K. And if you want to
But you get traveling waves.
you can make it oscillatory. Okay, then. Okay.
No. Then you go through. If you go through the hot bifurcation, you do the analysis around the hot bifurcation. You get a CD. You get a complex Greenberg-Landau equation, and then you get traveling waves.
But if you want to do Hopf bifurcation, then do binary fluid convection, and you have a finite K that's chosen from the beginning in a reflection symmetric domain.
Oh.
Anyway.
Well, it turned out what.
I'm sure it's
no.
an interesting system.
You're right. No, it turned. Yeah, it's just.
Of a large class.
Right. Okay. No, but this is the point. The interesting thing is that this is a new mechanism for generating a complex. This mechanism is a new generic mechanism for generating complex.
I don't doubt it's very
Okay.
interesting. I just
All right.
thought-
Yeah.
Let me, I wanted to, first of all, definitely there are systems
Okay,
which
yeah.
have this property. I would not go to detail. I have a question. Do you think, due to your classification and stressing the fact that you have both imaginary part and finite K is important, but still instability in your case, as far as I understand, appears where K equals 0?
No.
No, it appears.
No.
When you
No.
change
No.
parameter.
Okay. When you change. Okay. So. It's either or, because you're changing. What you're changing is the coefficient of K squared.
Yes, that's what.
Right. So if that coefficient is positive, you have a whole bunch. It's increasing parabolically. Right. If it's negative, then they're all negative. So if you have one, you have an infinite number. You don't ever have one. The only way to go through the instability is to change the system size. You can go through the system, but then, right? So if you take a system which is tiny, there's no instability. You make it bigger and bigger. You can make a system that only 1K there because that's, because the system is too small. But then you make the system bigger and you get more.
I wanted to ask a question. If you find instability which appears via finite case,
Yeah.
so it was fully stable, you change parameter, it appears via finite case,
Right? Yeah.
and additionally it has imaginary part. Would it be new in your classification?
No, we know it exists. The question is there a simple, what is the simplest kind of, we're looking for a standard model equation like, like a brusselator. Some simple, pedagogical example you write down and that you can see it happening before your very eyes in an analytic way, and
I don't think that 8th-order polynomial is not so simple for students, but this is...
No, but in the end, but the point is, in the end, it is simple.
It's 2 by 2.
It's a 2x2 system. That's the whole point. Otherwise,
It would be nice to write, a priori, a 2 by 2 system that would give.
Yeah, no, you can. No, all you have to do.
Or you don't care.
No, all you have to do is take demembrane you take d sol to infinity. If you take d sol to infinity, then in fact the solute fields are constant, and then everything is happening in the membrane fields, and it's a 2x2 system.
Okay, so you can implement this protocol at the level of the PDEs themselves.
Yeah.
Just getting a reduced.
Yeah.
You don't care about anything
Right,
else.
but this historically we just analyzed the system that
Okay,
was
I
given
think
to us.
We should go for lunch now.